questions with you, so just so you know. Ashley's going to be recording her own answers since she was gone yesterday, but. Okay. Yesterday in the first part of this case study, we talked about the genetic mechanisms that were working and kind of the modes of inheritance for the two traits that we were looking at, the body armor and pelvic spines in the sticklebacks. And we determined each of those traits was just controlled by one gene with two alleles. It was dominant to have those traits and recessive not to. And we went through some of the genetic experiments that they did to determine that. So we know this is a genetically determined trait, which means natural selection could be happening on these traits, and that could be why these freshwater sticklebacks have lost the body armor and pelvic spines. We are now going to kind of focus in specifically on the gene that controls the development of the pelvic spines. Body armor is probably a similar story, but we're just going to focus in on the pelvic spines for now. And we ended yesterday looking at this slide and kind of talking about how genes are expressed and the fact that in addition to the actual coding region, which on this diagram is kind of this purple section, there are also little bits of genetic information that are regulatory sequences. Think of those kind of like the parts that can turn this gene on and off that control when transcription of this gene happens. You're going to see several diagrams like this as we go through the rest of this case study today, so I want to make sure that that all kind of makes sense. Do you have any questions about what this diagram is showing? Okay, so scientists were able to actually identify the gene that controls the development of pelvic spines in these sticklebacks. And it's a gene they called PIDX1. I don't know why they named it that, but that's its name. And what they figured out is that PIDX1 actually doesn't directly make the proteins that the pelvic spines are built out of. It makes a protein that then goes out to the rest of the stickleback's DNA and bonds to the promoter region of several other genes. It acts as a transcription factor to initiate transcription of a bunch of other genes. So it's kind of like a master switch. It turns on a bunch of other genes, which then presumably go and make the proteins that the pelvic spines are built out of. Does that make sense? So PIDX1, when that is transcribed, builds pelvic spines. So this is not a question that you have to write down, but just turn and talk real quick. How would you expect this gene to compare in the marine and freshwater sticklebacks? Just turn and talk about that real quick. You don't need to write this one down. Nope, you're not writing this down. You're just talking about this one. Oh, yeah. Oh, gotcha. Oh, yeah. Okay, so again, the question is, how would you expect that PIDX1 gene to compare in marine and freshwater sticklebacks? Just talk about that real quickly. What would you expect to find here?
Find it in freshwater, but not in marine.
Other way around.
You mean the other way around? But I got the point. So scientists thought that might be true too. They actually went in and genetically sequenced all of the bases that make up the actual coding region of the PIDX1 gene. They were absolutely identical. Both freshwater and marine sticklebacks have this exact same piece of DNA. So that wasn't necessarily the difference. So if the coding regions are the same, how can we explain the difference? So what they did is they did another experiment here. that has to do with how those genes are read and kind of what happens. So when a gene is turned on, it's transcribed in a messenger RNA, right? Scientists can track where the messenger RNA is because they can actually use a dye that attaches only to those messenger RNA molecules that are produced by this gene. So it can show where this gene is turned on and where that messenger RNA is being made. So they did this to try and compare the patterns. And so as they were looking at these developing embryos in the fish, talk about this. Where do you think they will find messenger RNA from this gene in the marine sticklebacks? And where do you think they will find messenger RNA from this gene in the freshwater sticklebacks? Remember, now we're just focusing on the pelvic spines and the development of those. You don't need to worry about body armor anymore. But think about this. Where do you think they are going to actually find messenger RNA produced from this gene? That is your next question on your answer sheet. So turn and talk about this one and record your answer on the answer sheet. Are you ready to see what happened? So I'm going to show you what they actually found. I'm going to turn the lights off so you can see this picture a little more clearly. I'm going to go the other way to turn the lights off. So here are the results from that experiment. The regions that are stained kind of purplish are where they detected messenger RNA from the PIDX1 gene. So in the marine fish, the one on the left here is the marine one. This is the side view. This is the side view. You can see there's a bunch purplish here and a little spot of it here. This is a close-up of kind of the nose and jaw area. So you can see there's a dark spot of purple there, a bunch of it around there. And this is kind of a ventral view looking at the stomach side, and you can see there is messenger RNA there and there. So that's all the marine stickleback. The freshwater one on the right has it visible in the nose and jaw region, that front part. But you can see in the ventral view, there's none there, and there's none in these spots either. So your next question, What can you infer about the coding region of the PIDX1 gene and where it's being transcribed into messenger RNA? And did your prediction match the results of the experiment? Take a minute and answer question number 6.
Well, if you change yeah. Wait, what's this called again?
The gene?
No, the size of them.
The pelvic spines. Yep.
The pelvic spine. So we can further focus whatever is being transcribed. Write your answer. Is your prediction actually?
So what did you learn about this gene from this experiment? Yeah.
Like we know, like the whatever how you pronounce that gene would be transcribed like in the mouth because it has similar bone structure like of the spine.
Okay, so the part about the pelvic spines probably was not super surprising, right? This is being transcribed in the marine one, but not in the freshwater one. But you also discovered, is pelvic spines the only thing that this PIDX1 gene is responsible for? No, because it also showed up in kind of the nose and mouth region. So when scientists tried to explain this, they kind of came up with this diagram to explain this. We drew this already with these 3 pieces of regulatory DNA, but now they kind of label them. Think of the green circle as a regulatory piece of DNA that turns this gene on and off in the pelvic region of the fish. The red square turns it on and off in the nose, and the blue triangle turns it on and off in the jaw. What they think happened is there is a mutation in the regulatory DNA in the switch that controls this gene in the pelvis. We know that the regulatory pieces that control transcription in the olfactory region, that's the nose, and the jaw are not affected. How do we know this? That's your next question. Take a minute and answer question 7. So now scientists kind of had a genetic mechanism for how they think the genes changed. It wasn't the PIDX1 gene itself. That one's identical in both. It was the genetic switches, those pieces of regulatory DNA that turn this gene on and off. The switch that controls the activity of this gene in the jaw and the nose are fine because they could still detect the messenger RNA in both fish, so they know that one hasn't been affected. But the idea is that there was a mutation in the regulatory DNA that controls when this is expressed or how this gene is expressed in the pelvic region. So then they had to try and come up with the reason why. So now we have the genetic change. Now we need a reason why this trait might affect the fitness of these fish in freshwater and saltwater. So here are some things to know about the environment that these fish live in. Pelvic spines helped to prevent predatory fish from eating them in the ocean. In the ocean, the predatory fish would just bite the whole thing and they're spiky and they don't like it, and so they'd spit them out. So it helped them survive. But those predatory fish are not found in freshwater lakes. In freshwater lakes, you have different predators, which tend to be large insect larvae. That's what this picture on the right is. And those insect larvae actually grab onto those pelvic spines to eat the fish. So, based on that information, see if you can explain how this process of natural selection might have happened to result in a loss of these pelvic spines. That's question number 8. So what happened here, there was a relatively small genetic change, just a mutation in a piece of regulatory DNA, but it resulted in a fairly large morphological change, which means change to the body shape and body plan. This fish essentially completely lost its hind fins that it used to have. So think about the definitions we've learned about microevolution and macroevolution. Would you consider this microevolution or macroevolution, and why? There's a lot of debate about exactly where the line between those two is, so there's not necessarily a correct or a wrong answer here, but pick one and describe why you chose the one you did. All right, so this is a fairly small example, right? We're looking at little tiny fish that lost these little tiny spines, and this may not seem super important, but similar genetic mechanisms are at work in lots of organisms. And say in humans, if we lost the construction of our pelvis, that'd be pretty darn detrimental to our survival, right? Bad deal. That type of mutation is not known in humans. Scientists have, however, done other experiments where they did something similar to humans losing a pelvis, something like that in mice. These are pictures of mice embryos, and what they did here is they created what they called a knockout mouse. In this case, what they did is they deleted part of the coding region of the PIDX1 gene. So these mice can't make the PIDX1 protein at all. And so you can see there's some huge differences in the jaw region and in the hind limbs, especially, and probably a few other differences in the front limbs as well, but especially in the jaw and the hind limbs, which are the same places that we saw this gene active in the sticklebacks, right? It was in their mouth region and in their hind limbs. In this case, though, there's a really important difference between these two. And I think I want to go back and have you answer that question before I show you this next part. Let's see. I don't remember when it asks the question. There you go. Okay, so mice that don't have the PIDX1 gene, where part of the coding region was deleted, don't survive. Like you saw in that picture, they have defects in the head and jaw and the pelvis and the hind limbs. They hardly develop at all. 3-spine sticklebacks have completely lost the pelvis, but everything else looks totally normal. So explain that difference. The diagram they're referring to here is the one that shows the circle, square, triangle, and the coding region, that shape representation of this whole gene complex. So if you want as part of your answer, you can sketch kind of what that would look like for the knockout mouse. So how are these two cases different? Answer that in question number 10. This is an important point to understand for this case study. What's the big difference here?
With the mouse or mice, there was like multiple defects that were affected when like the gene was not coded for. Where like in the stickleback fish, the pelvic bone was just like taken away basically, but there was no other defects and every other region was functioning.
Okay, tell me more about that. Why?
I understood that because like all the defects in the mouse were necessary for the survival, like for them to have those, like the head, even the head, like need a fully developed head in order to survive. So, but the sticklebacks don't need their pelvic spine to survive. It was just an evolutionary adaptation that helps them to survive in the wild.
So why were there so many more defects in the mouse than there were in the sticklebacks? Brianna? Yeah, so one of the big differences and a really important thing to notice, let me see if I have this. Nope, we'll talk about it in a minute. So if I kind of recreate, I don't remember if all of these shapes and sizes are exactly right, but if we recreate this kind of thing, so we've got our regulatory sequences here, and then we've got our actual coding region of the gene, and this is all along our DNA strand. This is the actual gene that makes the protein. In the mouse experiment, in the knockout mouse, they deleted part of the actual gene. Nowhere in that entire mouse's body could it make the PIDX1 protein, which is why the nose didn't develop, the jaw didn't develop, the hind limbs didn't develop. Nothing that required that genetic switch, because remember, PIDX1 goes out and turns on a whole bunch of other genes. And so if it's not there, none of those other genes turn on. It has all kinds of defects, and that mouse can't survive because those defects are too severe for it to develop. A mutation in the actual coding region of the gene is almost always fatal if it's a gene that sets up major pieces of the body plan like this. But mutations in regulatory DNA, so in our sticklebacks, the mutation wasn't over here. The mutation was just in a piece of regulatory DNA, which can cause a pretty big change in what those fish look like. But it doesn't affect the entire expression of that gene throughout its entire body. It can still do its job in other important areas, like developing the nose and the jaw. It only affects that one part of the body. So those changes aren't necessarily as detrimental as actually knocking out the entire gene. Regulatory DNA and changes in regulatory DNA have been really, really important throughout the history of the evolution, especially of body plans and body shapes of animals. You might remember in 10th grade learning about Hox genes that control like where the head and the tail and the back and the front and the different segments of a body are. It kind of sets up the body plan. Mutations in those types of genes that control the shape and size and position of parts of the body can make what look like pretty dramatic changes in the shape of bodies from very small mutations. They can do things like change a neck from this nice short little human neck we have to a really long neck like a giraffe, not by actually changing the gene itself, but by changing how that gene is regulated. So the regulation of genes is super important in the process of evolution. And scientists have found that a lot of changes that look really huge actually boil down to a pretty small genetic change in regulatory DNA. This example that we see with sticklebacks, the loss of pelvic limbs or a pelvis itself has happened several times, like in snakes and in dolphins and in, I think that's a skink, and whales and manatees. They all show this same pattern. They've all lost their hind limbs, right? So now, based on what you learned from sticklebacks, make a hypothesis about how all of these animals may have lost their hind limbs through the process of natural selection. Again, you can use a gene diagram like I just drew, like we talked about earlier, to show kind of what you think happened and what you think this gene complex might look like in manatees. Take a minute and answer question number 11. So one really interesting piece of evidence that kind of supports this idea that there are mutations in the regulatory DNA and not the gene itself, if that's true, and if that's true in all of these creatures, that means the gene that's responsible for building hind limbs is still there, and it's still functioning in other parts of their body. And occasionally in some of these animals that have lost their hind limbs through evolution, a rare individual is born that has hind limbs. This is a dolphin that has hind flippers. Most dolphins do not. When a trait reappears like that, that has disappeared through evolution, that's called an atavism. That's the word they used for that. So, last piece of this to kind of complete the story, based on what you know, what do you think the genetic basis of the reappearance of these hind limbs might be? You can use another diagram, or you can just explain this in words. as you wrap this up, first of all, when you look in your notes from this unit, there are a couple slides. There are like 2 or 3 slides after this that we didn't get to. And that's okay. They just kind of expand on this idea of regulatory DNA and DNA that determines the body plan of individuals, and how mutations in those regulatory pieces of DNA and genetic switches is really important in the process of changing the appearance and the body plan and the shape of organisms. And that's a really important key in the process of speciation, especially looking at big changes throughout the history of life, like the loss of hind limbs or animals developing arms and legs instead of fins. Those types of things often have to do with the regulatory DNA. So finish up the questions that go along with this and turn in this case study, and then just spend some time tonight reviewing for the evolution test tomorrow.